Thanks for the introduction just now. And my name is Lisa Jia, and I'm an application scientist from Sophion Bioscience. Thank you for joining this webinar, and it's a great pleasure to be here to introduce our technique. First and foremost, I'd like to give a very brief introduction about our company. Sophion Bioscience is a Danish company which focuses on automated patch clamp. Currently we have different throughput products called Q-Patch Compact, Q-Patch, and Cube. And also we provide some different solutions, for instance, intracellular solution and extracellular solution. And also we have a collaboration with BCs. We can provide some stable lines. And of course, if you have you want to learn more information, you can visit our website here. It's showed below. And our headquarters is in Denmark, Copenhagen, and also we have different demo labs in US and in Japan. In South Korea, we have our distributor here, and also we have a collaborated demo lab in China. So currently for our partners, mainly from 3 different types. For big farmers, they own our higher throughput system, and a lot of CRO companies, they also own our system to help their customers to finish some projects. And also from some universities and scientific institutions, they own our system to do some very basic scientific studies. Here I showed our main products. And for our main solutions consist, of course, hardware and software. And we also have offer consumables here and also the accessories. For here, this is our main 3 different products. Here we separated by different throughputs. Here, Q-Patch Compact, we call it's a low throughput. We have 8 channels. One time you can get at least 8 cell recording, and we have a Q-Patch 2. This is already 2nd generation. And for Q-Patch 2, we have a 16 and 48 channels recording. So one time you can get 16 or 48 cells. And we have a highest throughput, which called the Cube. That means at the same time, you can get 384 recordings at the same time. So our technique mainly focus on, we focus on automated patch clamp just because like for manual patch clamp, it's a very, very high requirement technique. And for people, if you want to do the experiment, they will spend a lot of time on training, practice, and also still one time you just get 1 recording. So for the experiments, people have to spend a lot of time on it for the operation. Our system try to solve this problem. So we designed different throughputs to fit different requirements to improve the efficiency. And also you can try to get some faster drug screening. If you study like different compounds, you can measure a lot of compounds at the same time. But why we use the patch clamp, we have to come back to some very basic knowledge. So ion channel is a specific protein which crosses the cell membrane. And here, when you give some stimulus, there will be a current signal there. And most of fast activities will relate to ion channel changes. And here, this is some physiological rules for ion channels. So here first, ion channel will have an effect on intracellular signalings, and also ion channels could affect membrane potential regulation, and especially for brain, ion channel could also induce some synaptic transmission, and like for instance, for some other mechanism could affect secretion and absorption, and so on. And currently, for some studies have showed a lot of drug targets are on ion channels. From this figure, we can see it's almost 18% targets are about ion channels. And also not just the drug targets, also many disease mechanisms are related to ion channel changes. For instance, the most often studied pain, and also lung QT syndrome, heart arrhythmia, and recently more and more studies are related to other diseases for cancer and diabetes. So it's very important to know what happened with these ion channels. Then we have to use Patchclamp to look at the functioning change on ion channels. So to look at ion channels, there are different types, mainly 3 different kinds. So first of all, we know the one most well-known is voltage-gated ion channels. For instance, the sodium ion channel, potassium ion channel, and so on. And the second one is the ligand-gated ion channel, especially for neuron ion channels. And the third one is magnosensitive ion channel. Of course, there's some other kind. For magnosensitive ion channel, recently the very hot study is about piezo, just won the Nobel Prize. So here, to give more details about Patchclamp, as we know, Patchclamp has different model. One is voltage clamp, the other one is the current clamp. And look at the picture right side, you can see this is a mild side here, and this is a glass pipette. So for this recording, we use a current clamp to record action potential. And here, this is the stereo of a Patchclamp. Mainly is about we use a glass pipette, try to get approach to the seal. Then we change we give it some stimulus to measure the current changes or the membrane voltage changes. And here, this is the principle of the conventional Patchclamp, just like showed from this slide. And you can see here, this is a glass pipette and full of intracellular solution. And for outside, this is the extracellular solution. And we lay one cell here and try to measure the changes of the electrical signals from this cell, and this is the recording electrode, and this is the reference electrode. So this is the conventional Patchclamp, how it works. Compared to the conventional Patchclamp, our system is using a different Patchclamp way, which called a planar Patchclamp. And it's also not difficult to understand. To look at here, this is a conventional Patchclamp, and we use the pipette to try to approach the cell. And then now we put this pipette up and down, make it a planar. Then we use the gravity and pressure system, try to get this cell on this small hole. So here, this is our consumables internal structure. And you can see here, first we fill in the intracellular solution, and there here we fill in the extracellular solution, then we add in the cells. Then we start to give some stimulus to look at what will happen this cell, and then we will get a recording about the ion channels. So for our consumable here, there's a lot of advantages for the measurement. Because this is a very small volume. Every time if the cells are ready, if we want to measure some compounds, every time we just need to add in 5 microliter compound solution, and we can change all the time. And we have 155 waste space. So you will in total, we can wash in around 46 more or less different concentrations or compounds. So in general, you will not use out this kind of space. And also for here, we use the glass-coated material to avoid some sticky compounds. And we also built in the electrodes here and here. So you don't need to maintain your silver electrode, and the data in general will be very stable. And due to this design of this structure, we usually have a very stable recording. You will not lose your cell due to the shaking, due to the people walking around the equipment. So you can always start to try with a physical solution, which is so many experiments. They started with fluoride ion solutions to help get a better seal, and our solution could use the physical solution, which is much better for some experiments. And this is the ingredients of the different system. The left side is the manual patch clamp. The right side is our automated patch clamp. As you see, for manual patch clamp, they have different ingredients. You need to make the glass pipette by your own, and you need to have the microscope, the fridge cage, and anti-vibration table. So we usually ask for a higher requirement about this setup. And for our system, you just need to find a quiet place and place it there. So it's very easy to set. And also for the operation, for manual patch clamp in general, we ask for long-term training, because if you want to do manual patch clamp well, you have to practice a lot, and you have to understand everything behind. For the automated patch clamp, the biggest advantage and also our first purpose, just try to, okay, how can I start my experiment as fast as possible? So for all the operation, every protocol has been set there. So you just need to, for QPC, you just need to add in the solutions and the cells. That's it. Then you press the button, you start your experiments, and then you get your data, you analyze the data. For the rest of two equipments, you just need to place everything inside of the equipment, press the start. So everything is very easy to operate. You don't need to spend a lot of time to learn how to operate the system. Here I will give more details about how does our system work. And here, this is still the internal structure of our consumable. And when we add in the intracellular solution and the extracellular solution and then cells, we give a negative pressure, and also due to the gravity of the cell will lay on this small hole. And then we adapt the pressure to get a better seal, and we open this cell. After that, then the cell is ready to run the experiments. Then you can study your experiments. You can give different stimulus. For instance, you change the voltage. You give a ligand, like a compound, to study the ligand-gated ion channels. And also you can change the temperature and so on. All of these factors, it's possible to study. And then you play a current clamp or voltage clamp, you will get your results. So here, in summary, for our system, we will use a single cell suspension. So we always get some questions from the customers or potential customers if we can study brain slides or some tissues. Yeah. So I'm sorry to say, sorry, we cannot. We study the single cell suspension. So you have to isolate the cells to be single, and then you measure the changes of the ion channel on this cell membrane. But we can run whole cell recording and perform the recording, and Also, we have a much higher throughput for the measurement. For instance, this we can get 8 recording at the same time. For Patch 2, we can get 16 and 48 recording. And for Cube, it's a super high recording, about 384 data points. And also we have both the voltage clamp and the current clamp. And you can, if you try our system, you will know, okay, it's a very fast procedure to approach to a jiggle seal. And all the recordings, when you start the experiment, our system will record all the data points, and you will always you can always look back what happened with your experiments. And also we have the ready to use the electrodes you don't need to maintain, which saves a lot of time and energy for the people, and also especially for the new users, they don't need to struggling about all the maintenance. And all the setup you can connect to the temperature control. As we know, temperature has a very, very big effect on the ion channels. So we prefer to have a temperature control at a certain temperature. Then we will get a consistent data to get a better analysis and results. And also, just like I mentioned, we have a fluoride-free recording in a physical solution. So here, I will give a very straightforward show about our different throughputs. This is so-called Patch Compact, which we just released from 2022. It's a very short and our new star. And you can see here, we at the same time, we can record 8 cells. And for each cells, they are in parallel and independent. You can easily choose how many cells you want to record. Like for instance, I choose 4 of them. I run an experiment, and for the rest of 4, if I don't use for the consumables, I can left it for the next time use. So it's very flexible to use. And for Patch 2, we have 2 different versions. One is 16 recordings. The other one is 14/8 recordings. And for this system, it's fully automated patch clamp. And when you place everything inside, for instance, you can see here, this is a cell station. And here we can place some plates here. You just prepare the intracellular solution and the extracellular solution, get a flask of cell and do the isolation. The cells will be still in the single cell suspension with medium, but you don't need to wash your cells by yourself, and you place it here. The system will help you adapt the cells, the density of the cells, and then you set all your experiments and close the machine. You start. When you start, this system will not stop, but you can just walk away, do some other experiments. So you will save a lot of time for your study. And for our cube, this is a super, super high throughput. Like you have a 384 ion cells to record. You get a bunch of recording. And also for this system, it's very powerful. You can even study the light effect on the ion channels. And I will give more info how easy about this system to play. This example is about Patch Compact. So for Patch Compact, we call it a semi-automated patch clamp because we still need the person to play a little bit with this equipment. First, we need to know which we are going to study, which ion channel we are going to study. So we make a voltage protocol. Then the person need to use this pipette and in the solution in the cells. Then you start your protocol. You get the signal recording. After that, when you finish your experiment, you go to our software analyzer to analyze your data. So here For the person, they just need to follow the instruction from this system to run the experiments. And also for this system, we both have a voice reminder and a text reminder, everything to avoid some mistake by the person. And this is the workflow for how you get the data. You just need to insert the QPlate to the QPC, and then you add in the liquid, and then you add in the cell. When everything is started, you see the cells are ready to run experiments. Then you start your voltage protocol, you end your compound, and then you start your recording. After the experiments, you start to analyze your data. So it's a very easy way. You don't need to, you will avoid all troubleshootings about the manual Patchclamp, because for manual Patchclamp, probably you have to do the compensation by yourself. For our system, the system will do them all. And this is how it's flexible. Is it? You can say for our system here, one time you can run 8 sites at the same time, which means that you can record 8 cells, but you can also choose 4 of them here. And for each site, they are totally independent, and they will not affect to each other. So for the first site, you can run some voltage protocols and have a totally different design about the experiment. You can, for the second site, you can run another different protocols. So for 4 cells, you are already looking a lot of data, and you will get a lot of information from these recordings. It's very good for to play a trial or assay development, because for this data, you will get many information. And this is our Sophion Q patch. For Sophion Q patch, we have 16 and 48. It's very easy to operate. You just need to prepare the solution and the cells, place it here and here, then press the start. Here, every operation just with a touch screen. And this is the workflow about the Q-patch. And you can see here from our analyzer, you can easily edit your experiments, which kind of voltage protocol you want, and which kind of compound you are going to test, and which kind of concentration you will measure. So all of this information you will just design by yourself and make an assay with this system. Then you place everything inside the equipment, then you press start. Then the system will record all the data until they stop this experiment, and you will go back to the analyzer to analyze your data. And for Q-patch, actually, if you stop in the middle, this experiment will stop. So compared to the Q-patch compact, Q-patch 2 is more automated, but Q-patch compact will be more flexible. And we have tested a lot of cell type with our system. We did many stabilize with different ion channels, and also we currently we tried a lot of cell primary cells and iPS cells. Here I show some data here. As you see, we just pick one data randomly, and all of the figure we get from our analyzer. And analyzer is a very powerful software. You can easily edit your figures, and you can use it directly for your publication. And you can make IV curves. You can get your voltage protocols here. And also, you can also study all the activation and inactivations and so on. And for human iPS cells, actually, it's more and more popular, especially they have a lot of benefits for studies. They are from human. They are very good cell model to rule out the species specific. And also, you even can get some iPS cells from the patients and to study the mechanism of the disease. So for iPS cells, we tried a lot, but as we know, iPS cells is always difficult to operate due to a lot of factors. But our system, because of the high throughput, we always get a higher chance to get more information, and you can study the mechanism of the disease. You can screen the different cell lines, and also you can do a drug screening and so on. And here I show some information about our primary cell experiments. We also tested the primary cells with our system here, QPatch and Cube. As you see, we get the cell suspension from mice neurons or rat neurons. Then we place it in this system, and we do a high throughput screening. Because for the primary cells, after the isolation, they will be very weak, but still we will get, compared to the manual Patchclamp, we will get a lot of information from the high throughput equipments. And this is our results from our system. And you can see here, we can do a study on ion channels. For instance, we showed here potassium current and sodium currents. We can do a voltage clamp, and also we can do a current clamp recording here. We use the primary cells to measure action potentials, and also we can study the ligand-gated ion channels. Here we study the GABA ion channel, and also we measured some cardiac myocytes for on iPS cells. So for iPS cells, iPS-derived cardiomyocytes, as we know, it's very easy to be depolarized. So we want to rescue the depolarized cells by injecting the current. Previously, we used a standard current clamp to inject for the different cells where our program gave exact same current. Then for some cells, could be rescued, but for some cells, they will not get a very nice result. But currently, we updated this feature, and we use adaptive current clamp. We first gave a program to test how much current I should give for specific cells, and then for different cells, I will give a different current to rescue this action potential to get better data here. And also from our website, we have a lot of publications and application reports. If you guys who are interested in this kind of information, you are very welcome to check our Sophion websites. And I think this is all the slides I'd like to talk about today. And thanks for your attention. I think we have some time to get some questions, and I'd like to take any question of them.